morning, everyone. This is our laboratory report entitled Microscopic Observation of Mitosis in Onion Root. I am Fulhimm M. Pareja, and I will discuss the introduction. So for this activity, our group observed onion root tip cells under a compound microscope to study the stages of mitosis. During our observation, we were able to identify cells in early prophase. However, we did not observe metaphase, anaphase, or telophase. We believe this limitation occurred because we may have taken our sample from the wrong section of the onion root. The samples we examined likely had low mitotic activity, making it difficult to observe the stages of mitosis.
My name is Deb Collin A, Oxina, and I am here to present the objective of this activity. It is to identify and differentiate the various stages of mitosis, such as prophase, metaphase, anaphase, and telophase in onion root tip cells by observing their distinct chromosomal arrangement and cellular structures under the microscope.
Meg Ayessa Domiquel and I will be discussing the methodology. First is we prepare the dissecting kit for peeling and cutting the onion, preparing the glass slides and cover slip, the onion with grow roots, 5 ml beaker, clean and dry pipette, acetylcarmine solution, and providine iodine solution. Next slide, please. Next is we use proper PPE and we prepare the microscope. We cut the root tip of the onion. We put the root tip on the slide, put the solution on the slide, and put the cover slip on the slide. The next part of the methodology, Clinton will be discussing.
Good morning, I am Clinton Aron and I will be discussing the next slide of this methodology. Put the slide on the stage of the microscope, then examine the specimen of the sample of our onion root. Next slide. Cut the root tip of the onion root, put the root tip on the slide, put the acetylcarmine on the slide, and next slide. Put the cover slip on top of the slide, put the slide on the stage, then examine the sample of our onion root. This is the view of onion root under scanner, under LPO, and under HPO. Next slide, please. Results and discussion is for Aldrin Nobleza.
Good morning, I am Aldrin Nobleza, and I will be discussing the results and discussion. So the root tip is the best place to observe the mitosis because it contains the meristematic region, where cells are actively and continuously dividing. This region drives the growth of the root, so it normally shows all the stages of mitosis, the prophase, metaphase, anaphase, and also the telophase. So however, as you can see in this image, the only mitotic stage that we were able to observe clearly was the prophase. So our professor also confirmed that the cell we identified is indeed in the prophase stage. So the reason we observed only this stage, as mentioned earlier by my groupmate in the introduction, is because we accidentally cut the wrong section of the root tip, and also because this section contains fewer actively dividing cells. Even though we examined multiple regions of the onion root tip, the only stage we consistently found was anaphase. I mean prophase. So in this next slide, as you can see, the image under high power objective in the left corner, and we process the image using the image J. So we used image J because it is an easy to use software for analyzing microscopic images. And just like this one, it allows us to make accurate measurements such as the size of each cells, the cell wall as you can see, and the prophase, and also the number of cells in each mitotic stage. So we can see that there is only one mitotic stage that we can see in the image J. And it also makes the cell structure and particles easier to see. So this helped us verify whether any other mitotic stages were present in our sample, but again, only prophase appeared. So next slide. So in this section, we also compared our results to a reference image from the study of Ramsay Brett. So in this reference image, the stages of mitosis are clearly visible and very well observed because their specimen came from correct meristematic region. When we compare it to our own, we can clearly see the difference in visibility, the completeness, and also the completeness of the mitotic stages. So the conclusion will be discussed by Mr. Ryan.
Good morning. I will be discuss. I am Ryan A. Elopre. I will be discuss view of conclusion. Conclusion. To sum up, students learn how to properly prepare for microscope activity and the microscopic activity of the grow germinated onion roots allowed for the clear observation of actively dividing cell in the root tip. In this activity, students learn how to observe the different stage of mitosis and allow them for a clear observation of how cells activity dividing.
Overall, for this laboratory activity,
Student also
the students allow them how to identify each stages of mitosis using microscope and the difference between using iodine solution and acetocarmine. Additional documentation about our lab activities. As you can see, we're all helping each other to to accomplish our task. Next slide. So here's the reference of our activity that you can visit for confirmation of our work. Next. And so that's all for our visual presentation for asynchronous class. Thank you for listening. Good morning.